Before I started my PhD on the GSK Strathclyde Collaborative PhD programme, I did an integrated MChem degree at Oxford University. My master's project was in spin chemistry, so quite different from where I ended up with my PhD work. And I then moved on to the PhD programme, and the rest is history. My PhD project sat sort of between medicinal chemistry and chemical biology and was based at the GSK Stevenage site. I was working on bifunctional small molecules, including antibody recruiting molecules for targeted cell killing and lysosome targeting chimeras for targeted protein degradation. My project began with a lot of synthesis, but I did do an increasing amount of biological assay work as I made my way through the project, and I picked up new skills. And by the end of the PhD, I was running cell-based assays, and I was fortunate enough to learn some of the skills for that during a secondment to the GSK Upper Providence site in the US. As I mentioned, the PhD was quite a big change from my master's project, and it was certainly a steep learning curve and a challenge when I started. And the reason for the change from my master's to the PhD and moving into the pharmaceutical industry was it was only very late on in my degree that we had any exposure really to medicinal chemistry. And I remember the lecturer explaining the kind of design and mechanism of cisplatin, and it just got me really interested in the idea of rational drug design. And so when I saw the opportunity to learn more within the pharmaceutical industry, that was really appealing to me. I also knew that I didn't want to stay in academia long term, so I thought it was a great chance to get some relevant experience in industry and of a work environment more generally, at the same time as taking on an academic project as a PhD. But at the time I wasn't really looking for PhDs in academia, and if I hadn't ended up on this particular program, I think I would have probably worked in research and development and then thought about further study later on in my career. So during COVID, the PhD students based at Stevenage and in my cohort, they spent quite a lot of time out of the lab or on a rota with more limited lab time to make space for essential project work within the kind of socially distanced system that was implemented. I was fortunate in that I had run some DNA-encoded library selections shortly before the pandemic began, and so I was able to spend quite a lot of time running data analysis at home. But one of the kind of unfortunate impacts was that I wasn't able to get up to Glasgow for a secondment at the University of Strathclyde, which normally PhDs on the programme will do for 3 months or so. And I would have really liked to get some experience of a different type of chemistry and to get to know my academic supervisors group, and really spend some time in Glasgow would have been great. So that was a bit of a shame. And the other thing that I think we all missed out on was in-person conferences. I was lucky to be able to attend quite a few virtual events, which I really enjoyed, and I like presenting and communicating my science to other people, but it would have been really nice to present more in person and kind of connect with people in a more meaningful way. So I always find the question of where I would like to be later on in my career quite a tricky one. I never feel like I'm planning things out very far in advance, but I would like to stay in drug discovery in the future, and it may sound very cliche, but I would like to feel that I'm making a contribution to getting drugs to the market, which will really make a difference to patients. When I finished the PhD recently, I was not very sure which direction I wanted to go in terms of the types of role to look for. I have just taken a job in clinical development on that side, which will be a big change and again a lot to learn after the PhD. But I'm really looking forward to starting and seeing what I make of that new side of drug discovery.